Hello and welcome to this module 3 of this basics of immunology course. In this module, we'll discuss two important things. One is hypersensitivity reaction, another one is complement cascade pathway. The hypersensitivity reaction is basically the abnormal physiological condition where some undesirable and adverse immune response towards antigen occurred. So there are four different types of hypersensitivity reaction: type 1, 2, 3 and 4. So this is type 1. Type 1 means this is IgE mediated hypersensitivity. So IgE or immunoglobulin E is the main factor in this type of hypersensitivity. In type 2, IgG or immunoglobulin G is main component for the hypersensitivity reaction. In case of type 3, there is formation of antigen antibody complex. So immune complex are formed, that means antigen antibody complexes are formed, and they are being recognized by the neutrophils, which are kind of one type of white blood cell, and this kind of activation occur in case of type 3 hypersensitivity. In case of type 4 hypersensitivity, this is also known as cell mediated hypersensitivity. Here antigen will get bound with that antibody which is present on the antigen presenting cell, and they will secrete some chemical modulators which are also known as cytokinins or cytokines. These cytokines will activate both the macrophage and the cytotoxic T cell, so that the cytotoxic T cell can produce some T cell mediated immunogenic response, and the activated macrophage can have some engulfing effect on that affected cell. So this is hypersensitivity reaction. The type 1 hypersensitivity is also known as the fastest anaphylactic reactions. The major example is when some honeybee cuts our skin, there is a granular formation occur and it become very inflamed and red. So that is the type 1 hypersensitivity reaction, and that is mediated by IgE, because IgE is associated with the histamine formation or all the allergen and allergy type of condition that is due to this type 1 reaction. And the type 4 reaction is basically, it takes a huge amount of time, like Montu test. This is one good example for the cell mediated hypersensitivity, and it takes around 2 to 3 days for showing the response. So that's why this is also known as delayed type of hypersensitivity. The next thing we'll see is complement fixation. So complement system is some proteinaceous factor which are being activated in a cascading mechanism when some antigen interacts with antibody, and they can attack the pathogen cell membrane of the pathogen through membrane attack complex, and they can produce a pore or hole in the membrane of that bacteria, and it can destroy the bacteria. So this complement system should get activated. Now there are three pathways for complement activation. One is the classical pathway, another one is the alternative pathway, and third one is the lectin pathway. So the initial phase of these pathways are different, but ultimately they are, the last part of these pathways are same. So in case of classical pathway, when antigen antibody reaction has done, the C1 component. Now before that, let me discuss about the complement components. So there are 9 components, C1 to C9. So these 9 components are there, and C1 component can again be divided into C1q, C1r and C1s. So whenever in classical pathway, whenever antigen antibody complexes are formed, the C1q, R and S, they will get activated. They will activate the C4 component, and this C4 component will activate C2 component. So the C2 will be divided into C2a and C2b. The C2a will join with the C4, and they will produce one component, C4, C2a and C3b, and after that they will activate the C5. They will that will be a multi subunit complex, and after that all C6, C7, C8 and C9, all of them will be joined. They will form the MAC or membrane attack complex. They will attack the membrane of that pathogen and they will neutralize that pathogen. Okay. The second pathway is lectin pathway, where mannose binding ligand or mannose binding protein, they are mainly the trigger of this lectin pathway. So again C4 will be activated, and it will activate the C2, and ultimately C4b, C2a complex will be there, and that will activate the C3. Now C3 will again be divided into C3A and C3B, that will activate C5, and then again C6 to C9, all those complexes will be there, so that will produce the MAC complex, and they will attack the membrane pathogenic membrane. The third one is alternative pathway. Here, if there is any the surface of the organ is activated by some antigen presenting cell or so, then there will be one factor that is known as factor B. It will come and it will bind with the C3 component, and it will form C3B. C3B means component 3 has two parts, C3A, C3B. So this C3B plus that B factor. Again with that B factor, some D factor will come, some P factor will come, and it will ultimately produce one big factor. That big factor is also known as factor I or factor H, and that big factor will automatically update or activate the C5. And once C5 is activated and attached to that factor I or H, they will add all this C6, 7, 8 and 9 to them. Again they will produce the MAC complex, and that MAC complex will attack the membrane of the pathogen. They will produce a hole, they will produce a pore in that in that membrane of pathogen membrane, and the pathogen will get neutralized. So this is the complement fixation pathway, and with this the module 3 ends. Thank you.